There we go. Hello, judges. My name is Benjamin Persily, and today I will be talking about how I utilized and optimized the prime editing system in two different types of pluripotent stem cells. Prime editing is a newer and safer version of the well-known CRISPR-Cas9 system and is composed of two primary parts. First, on the left, is the prime editing guide RNA, or pegRNA, and this is the component that my project is centered on. PEG-RNAs have three subcomponents: the primer binding site, which binds the primer in the target DNA, the reverse transcriptase template, which contains the complementary RNA sequence to the desired edit, and thirdly, what I focused on, the guide or spacer component. And this is what brings the entire prime editing system to the target. The second major component in this system is the prime editor, which is made of two subunits. The reverse transcriptase, which reverse transcribes that reverse transcriptase template, thereby incorporating the desired sequence into the DNA. And secondly, is the Cas9 nickase. And this, along with the reverse transcriptase process, is what sets prime editing apart from CRISPR, because CRISPR uses a wild-type Cas9, which employs a double-stranded break in the DNA, leading to higher rates of unwanted mutations, but the Cas9 nickase only nicks one strand of the DNA. These two components make up what's known as the PE2 system. However, I also use something called the PE3 system. The difference being the PE3 system uses an additional single guide RNA that has been shown to raise editing efficiency. I employed this prime editing strategy in two different genes. First, the cystic fibrosis transmembrane conductance regulator gene, or CFTR, and the SOX2 gene. In the former, I worked to both induce and correct the leading cause of cystic fibrosis. In fact, 70 to 90% of cystic fibrosis cases are caused by this three base pair deletion here. The latter, SOX2, I used as a model for the optimization of the prime editing system, the strategy for which I will discuss later. In my project, I used two different types of pluripotent stem cells. On the left are induced pluripotent stem cells, or iPSCs. To make iPSCs, somatic cells are derived from a patient. Then, via send-out transduction, are reprogrammed to be pluripotent, and these cells can later be differentiated into any cell type. The second cell type I used was a modified H1 stem cell line that had a TD tomato reporter knocked into the end of the SOX2 gene. And this process of creating this H1 reporter line was done before my arrival in the lab. In order to reprogram these fibroblasts into iPSCs, they were infected using send-out transduction of the four defined transcription factors for pluripotency: OCT34, SOX2, KLF4, and CMEC. They were then maintained in a feeder culture system before being transferred to feeder-free wells. Pluripotency was then measured by a flow cytometry, and three factors were measured for: OCT34, SOX2, and NANOG, another protein shown to indicate pluripotency. Down here, I have flow cytometry data indicative of a successful reprogramming, as 96.7% of the cells were positive for both NANOG and OCT34, and 98.3% of the cells were positive for both OCT34 and SOX2. Since the pluripotency indicators were present in almost all of the cells, these once fibroblasts were now concluded to be iPSC, and these stains above also support the pluripotency of the cells. The blue stain shows DNA presence, the green stain, OCT34, and the faint and fuzzy red indicates no remnants of a Sendai virus. If it was a clear and bright red, then the cells would need to be passaged further, but as you can see here, there are no remnants of the Sendai virus detectable. Subsequently, I cloned three pegRNAs for the prime editing of the CFTR gene. To clone them, I digested a backbone, and then four primers were annealed together. Then, these primers were assembled using PCR with that backbone, and the assembled pegRNAs were then transformed, and select colonies were screened and MIDI prepped. The accuracy of these pegRNAs was then confirmed by Sanger sequencing. These results here indicate the successful cloning of the pegRNAs. And the reason there are three of everything is because on the left is a pegRNA made to induce the mutation, in the middle is one made to correct it, and on the right is a new ePegRNA, or engineered pegRNA, also made to correct the mutation. ePegRNAs have been shown to improve efficiency because of an additional 3' structural motif. So I wanted to compare it against regular pegRNAs for the study of the CFTR gene. These gels up here represent a successful digestion. These plates, a successful transformation. And these gels up here, a successful colony screening. This sequencing down here shows that in the pegRNA to induce the mutation, the 3 base pair sequence present in the corrected versions of the gene of the PEG RNA is gone. These electropherograms therefore confirm the accuracy of the cloning of the PEG RNAs. To actually conduct the prime editing, the PEG RNAs and other prime editing components were nucleoaffected into the iPSCs, and these cells were sent off to the MiSeq deep sequencing system by Illumina to measure the efficiency of the gene editing, and the data was interpreted using the CRISPResso web tool for deep sequencing analysis. In terms of the prime editing of the cells, the efficiency of correcting the mutation was much higher than that of its induction. The induction of the mutation had very low efficiency in modifying the cell's genes, and none of the cells were shown to exhibit the proper CFTR induction. For the correction of the mutation, I used one of the simplest prime editors, which, as I mentioned before, was just the PE2 system as a control, and I paired this with a standard PEG RNA. And I compared it with several groups. The first group was the PE4 MAX system with an ePEG RNA. The PE4 system functions in the same way as PE2. However, an MLH1-DD protein is co-transfected, which is a DNA repair protein shown to improve efficiency. And the MAX system uses a modified prime editor, also shown to improve editing efficiency. The next group I used was the PE4 MAX system with an ePEG RNA and P53DD, which reduces cell death after nucleoaffection, thereby furthering editing efficiency. And the final group uses the PE5 MAX system with an ePEG RNA and P53DD, and it functions just like the previous group. However, an additional guide RNA is added to improve editing efficiency. In essence, PE5 is to PE4 what PE3 is to PE2. And overall, with these modifications, the final group's editing efficiency reached nearly 0.1%, which is a vast improvement of the approximately 0.01% of the original PE2 group. Now that I've discussed how I use prime editing to induce and correct the CFTR F508del mutation in iPSCs, I'm going to talk about how using the H1SOX2 TD tomato cells, I optimize the prime editing system. So, as I mentioned, I use a modified H1 stem cell line that had this TD tomato reporter knocked into the end of the SOX2 gene, part of which is pictured here. The 10 gray arrows that you see each represent a spacer target for a PEG RNA for a total of 10 PEG RNAs. And each PEG RNA would insert a precise TGA stop codon into the SOX2, thereby silencing the TD tomato reporter downstream. So, I nucleoaffected the prime editing components into the cells and then used flow cytometry once again to measure the editing efficiency. And if a cell had TD tomato turned off, then it was deemed successful. The data of the PE2 and PE3 systems demonstrated the findings that targets 1 and 7 had the highest editing efficiency, indicating that the TGA codon was properly inserted into the DNA. The reason that targets 2, 6, 9, and 10 were not tested in this experiment is because prior to my arrival in the lab, the PEG RNAs were cloned at too low of a concentration for the prime editing. So I took it upon myself to reclone them for the following experiment. For these data, I used the PE2 and PE3 MAX systems, which, as I mentioned, the MAX system uses a modified version of the prime editor shown to improve efficiency. Once again, targets number 1 and 7 showed very high levels of efficiency, and after recloning the missing PEG RNAs to a higher concentration, I found that target number 2 showed especially low efficiency. However, as you can see, the PE3 in either experiment, indicated by the red bars, did not substantially improve efficiency as it was expected to. To understand why, I conducted an experiment using only the additional guide RNA of the PE3 system, without the PEG RNAs. And as you can see, it had pretty low editing efficiency overall, thus explaining why the additional guide RNA did not substantially improve editing efficiency. Here, I have the sequences for spacers 1, 7, and 2. As you can see, spacers 1 and 7, the two highest performing PEG RNAs, both have 100% GC content at the 3' end of the spacer. However, PEG RNA number 2 had 100% GC content at the 5' end of the spacer. And the reason that I have it pictured next to target number 1 is to show that it's not about the locus within the gene that affects editing efficiency, but rather the sequence of the spacer. Therefore, I was able to conclude that high GC content in the 3' end of the PEG RNA spacer leads to the highest editing efficiency, whereas if the GC concentration in the 5' end of the spacer is high, this results in low editing efficiency. Overall, my project is focused on the combination of two extremely powerful biomedical tools. Initially, I demonstrated the potential of using prime editing in pluripotent stem cells by working to both induce and correct the lead cause of cystic fibrosis. The CFTR gene codes for the epithelial chloride transport channel protein. The mutation results in thickened mucus, which can block airways, inhibit functional proteins, and trap pathogens, leading to an increased rate of infection and decreasing the average lifespan of patients to only 47 years old. So, by correcting said mutation, the protein would have restored functionality and help the patient significantly. Following this experiment, I took a step back, working to optimize the prime editing system as a whole by uncovering the trend that high GC content in the 3' end of the spacer of the PEG RNA leads to the highest editing efficiency. And it's important to emphasize that not much work in gene editing is focused on the 3' end of DNA and RNA, as promoters and other important features of the DNA and RNA are found mostly at the 5' end, leading to more focus in research. However, my work in optimizing prime editing highlights the importance and largely untapped potential of the 3' end. The 3' structural motif of the ePEG RNA that I mentioned previously also corroborates the idea that more work needs to be done to truly understand the importance and power of the 3' end in the PEG RNA design. I also want to note that the reason why I chose to optimize prime editing over the well-known CRISPR system is that although CRISPR generally has higher editing efficiency, prime editing is far safer because it has far fewer off-target mutations, an extremely important quality to have when discussing the utilization of gene editing technologies in humans. So, if the efficiency can be raised, with prime editing being the safer option, it would become the de facto gene editing tool, replacing the more dangerous CRISPR-Cas9 system. Additionally, prime editing has the ability to correct up to 89% of disease-causing variants. So, this optimization has profound implications for gene editing's use as a tool for curing disease, because optimizing design for a critical component of gene editing will help further disease-focused research at an accelerated rate, helping more patients with many diseases faster. I also want to note that part of my data is currently serving as the basis for the work being done in the lab where I conducted this research in their efforts to develop new prime editing systems, and I'll be joining them and working on this project later in the year.
That's
time. That's time. Sorry.
Yep. Thank you, Benjamin. We'll now go directly into the 6 minutes of Q&A. We appreciate your